It's a very special pleasure to introduce my colleague, Dr. Richard Hynes, who's this year's Hope Funds for Cancer Research recipient of the award in the category of Basic Science. Dr. Hynes is a Howard Hughes Medical Institute professor and the Daniel K. Ludwig Professor for Cancer Research at the Koch Institute for Integrative Cancer Research at MIT. Cells are the basic unit of life. Most of us are composed of about 30 trillion of these things, and they stick together. And Richard's the only person in the room who understands how that works. His research focuses on cell adhesion and the interactions between cells and something called the extracellular matrix. And the interactions, there are problems in metastatic cancer. He's well known as co-discoverer of fibronectin molecules, a discovery that's been listed by Thomson Scientific Science Watch as a Nobel Prize candidate. Dr. Hynes has served the scientific community extensively through his roles as head of the MIT Biology Department. I believe he got me tenure. Director of the MIT Center for Cancer Research. He's been president of the American Society for Cell Biology, and he's a member of the Board of Governors of the Wellcome Trust. Most special to me, Hynes has nurtured a very special environment at MIT that places high value on individuals and their ideas, on scientific excellence, on education, and on mentorship. Dr. Hynes is a member of the National Academy of Sciences and the National Academy of Medicine, and a fellow of the Royal Society, the American Academy of Arts and Sciences, the American Association of Cancer Research. He's won many prestigious awards, among them the Canada Gardner International Award in 1997, the E.B. Wilson Medal in 2007, the David Rohl Medal in 2017, and the Albert Lasker Award for Basic Medical Research in 2022. Please join me in congratulating Dr. Richard Hynes on receiving the Hope Funds for Cancer Research Award in the category of Basic Science.
Well, thank you very much for the introduction, a very nice one. And thank you for the award of this prestigious award. It's been a delight to be here. And also, I was very struck by the importance of the way in which the Hope Funds support junior research scientists. That's the best way to spend money. They're the future. And the way in which you train them, support them, mentor them, and build a community for them to grow up in is very impressive. I was very impressed by the science presentations today. So thank you for doing all of that, and thank you for being here tonight. So, what should I tell you? Well, I've been a faculty member at MIT for 50 years, and I hired Rick, and it's true that we did get him tenure. Amazing. So, I was actually a graduate student at MIT. I came over to the States to be a graduate student at MIT because I wanted to try out the States and see a different scientific culture. I have a degree in biochemistry from Cambridge, but I wanted to see something different, and MIT was recommended to me, and that was a great recommendation. I came here as a graduate student, and I loved it. And I went back to England, which had been our intention. My wife, who was here, and I had got married, and we came to MIT 2 or 3 weeks later. So our intention was to go back to England, which we did. But then 3 or 4 years later, MIT offered me a job, so I went back because I knew that I missed the scientific culture at MIT. It was a very good decision because it still is and has been all that time a wonderful place to do science. And it shares many of the features that show up in your fellowship program. So as a postdoc, as I said, I went back to London, and as a postdoc, I became interested in cancer. I wasn't disinterested before that, but I became more interested. I've always had a strong interest in developmental biology, as have quite a few of your fellows, I was interested to hear. And I've always thought of cancer as aberrant development. It involves many of the same features as development, but just a bit disorganized. And that's the way I think about it. So when I was at a postdoc in London, I tried to find out some of the ways in which cancer cells and other cells share properties. And I was trying to find out how cells interacted with one another. This was in the early 70s. And we didn't know much at all about how cells interacted with each other, how they knew who to stick to, how they knew where to walk, where to stop, who to pick up as fellows in the activity that they were doing. And so I was trying to find out about cell surface receptors that mediate cell adhesion and might be distorted in cancer. And that actually worked. We didn't find cell surface receptors at that point. We found a big molecule from the extracellular matrix, which is the scaffold upon which cells sit. And it does a lot of things for cells, which I don't have time to tell you, but I'll tell you a couple. So we found this protein fibronectin, which turns out actually to be a family of alternatively spliced proteins. It wasn't a membrane protein, it was a matrix protein, a big fibrillar protein that makes fibrils. So the fibronectin-rich extracellular matrix fibrils, it soon became clear, lined up with the actin cytoskeleton fibers inside the cells. There was a very close overlay of those. And if you manipulated things, the fibronectin organized the actin filaments, and conversely, the actin organized the fibronectin. So there was obviously a connection between the cytoskeleton bones and the muscles inside and the extracellular matrix outside. So that became a golden prize that we wanted to find. So we went back to looking for receptors, and this time we found them. They're called integrins. And we and others have spent the last 50 years working on them because they play key roles in all of those processes, particularly metastasis, which I've always been very interested in. That's the process that kills many cancer patients, and we don't understand it as well as we should, and we don't quite know how to stop it, although we've now founded a company that's hoping to work out how to do that. We'll see. So that was a big change in what we were doing. We were working on matrix and cell adhesion instead of what we'd been working on before. And that's set the stage for everything we've done since. It turns out that integrins are on essentially every cell in the body, and they mediate all those processes, who sticks to whom, whether they stick to the matrix or not, how they migrate. And they do that in all metazoa. And in fact, you can make an argument that they were key to the origin of metazoa. That's how cell adhesion that produces multicellular organisms gets set up by molecules like integrins. So it's been an amazing trip the last 50 years following all of this stuff. There's lots more to do. But as others have said, doing science is such a trip. And it's so engaging that you want to go to work every day. And despite what one's family may think, we do go to work every day. So it's been a trip. So I want to say a couple of other things for the fellows. It's very enriching to do things in academia. You have the opportunity to do things outside your own research. You talk to your colleagues and you get interested in what they do, and that's very important, to be influenced by them, and I have been. But it's also important to do things like contribute to the organization of your department and your institution to establish the environment in which you work. And I've done a fair amount of that. But I think even more important, I want to say something about the effort that you can put into interacting widely outside. Running societies like the American Cancer Society, both Lainey and I were president of the American Society for Cell Biology, and that's a very enriching thing and a contribution to the society and a lot of fun. It led me into things which I'd never expected to get into. As I said, I'm interested in development, and I got involved through the ASCB in defending stem cell research during the Bush administration, which was trying to ban it, human stem cell research. And that led me into co-chairing a National Academy Committee to set up the rules that one should follow in doing that in an ethical way. And then a bit later, since that worked, for doing the same thing for human genome editing. And those were extremely enriching experiences. Collaborating with lawyers, which I didn't know much about, and politicians, which I knew less about, in working out how to do things properly and how the government works. At that time it worked. And so don't shy away from those things, not at the beginning of your career, because they take time, but later on they'll feed back in many ways, and you can influence things way beyond what you do yourself, which is also rewarding, along with the work you do and the pride you can have in your trainees, which we all do, and you've heard others say so. So finally, in closing, I want to thank all my mentees, graduate students and postdocs, and all my colleagues who've helped, and my competitors who've helped. Competitors are good to have, actually. They stop you going down blind alleys and staying down them, so because they picked the right choice and you didn't at some point. And finally, I want to acknowledge my family and my wife, who's down there, who's supported me through all of this period and provided a wonderful support for what we've done in the lab, built a wonderful family, and been a great supporter. Thank you to her and to everybody else.